talk is kind of, it's not two complaints, it's one story and one complaint. The story is that I'm trying to summarize the chapter that Willie and I have in the Tom Taylor Memorial book that's coming out shortly, right? And the second is a complaint about molecular clocks and phylogeny. This is a quote from Morris et al., Jenny Morris's paper, PNAS paper from earlier this year, which appear to show a Cambrian origin to the embryophytes, but also at the same time to kind of diss the fossil record of plant-like remains that are earlier than the middle Ordovician. So people seem to accept the tetrahedral tetrads as representative, as being an apomorphy of embryophytes, but everything else below that is considered problematic. And my argument is, and this is the argument in the paper, is that if you go back to Bauer and look at and his arguments about the assembly of the plant, his arguments about the antithetic origin of the sporophyte, you can use the arguments that he created from morphology and consider that the fossil record in regard of spores before sporophyte, that is, sexual phases occurring before vegetative sporophyte phase, and see if we can place that into a concept, a paleobotanical method of looking at novel characters in the fossil record and recognizing cells and tissue types, putting them in their stratigraphic order, and then the method would then be if we can have or try to infer what the developmental basis is for the generation of those characters, right? And at that point, we can make an assumption about whether these characters are co-opted from algal features that existed before in the streptophytic algae and the ancestors of the land plants, or whether these are novel structures or novel homologs that require a new evolution of a genomic component. All of that then is assembled. I use the term module in quotes. There's different words for what I'm trying to express there, but the idea is that you have to have some kind of assembly of the plant genome that occurs in time, and my argument now is that that assembly was completed by the time we have an axial, upright, sporophytic land plant in the fossil record, which would be somewhere in the Silurian and not in the Cambrian. So here are the characters that I'll be discussing today. These are based on things that are physically found in the rock record, all right, at various points. So we work on characters, so lamellated spore walls, which are considered to be plesiomorphic to the embryophytes. And what does that represent in terms of a physiology, or in terms of a developmental program that creates that character? And that is the deposition of sporopollenin from an algal zygote wall onto the haploid phase, or onto the spore phase. That turns out to be a little more complicated. It's not just a simple issue, but I'm going to skip over a lot of these things. I just want to sort of touch on the ideas of the little pieces of the fragmentary record that exists prior to the Silurian or prior to the Devonian that could possibly belong to this story. The occurrence of tetrahedral tetrads, in my view, this represents the canalization of meiosis. Meiosis before the middle Ordovician is messy, and so most paleobotanists recognize the tetrahedral tetrad feature. They tag that to the origin of the embryophytes. All right? I tag it to the origin of meiosis in plants, not the origin of the embryo per se, or not the completion of the embryophyte genome. This is Willie's thing. We have homogeneous spore walls in the Darwinian, and so his argument is that those homogeneous walls have to have a tapedal origin, not an endogenous origin in which the spore pollen is coming from inside the spore. Now we have a bunch of other things. Cuticle. I don't really want to talk about cuticles. Free trilete spores. So what happens in the record is that you've got something called imperfecto triletes, which is a broken out of the tetrad. Right? And that you get these torn proximal surfaces and blah, blah, blah. So it's kind of significant when we actually have true trilete spores, because to get a true trilete spore, at least at sort of in terms of bryophytic level sporogenesis, you have to have both kalos deposited between the developing myospores of the spore site, and then also a kalase function to remove that kalos that allows the spores to float free. So that having free trilete spores gives you a couple of clues about specific aspects of development within the added into the plant genome. Here's another one that's kind of annoying. The tubes that we start finding in the Silurian, they've all been sort of now dumped into the fungi, because the paleobotanists that study these things think of them as, you could sit down if you want. It's okay. Think of them as fungal, and in my view, the characters that make these fungal there really are no good characters. They're just tubes. All right, they could equally be, protonemata without crosswalls or something. I mean, there could be other origins to these tubes. And then the last part of this argument would be, well, that there's something with the tubes, if they are related to wood, woody tissue, that lignin synthesis is something in and then to make a vascular bundle, you have to take those tubes and glue them together. All right, that might happen just developmentally in terms of where the meristem is producing that tissue, but at some point they have to stick together, and so that's a cell-cell adhesion story. Which that's in the animal world. So that's the talk, and then here's a few pictures to describe that with a timeline. I'm sure there'll be no time for questions. I'm not going to go into the details of why I have these times where they are, but this is just my interpretation of where these particular characters are first seen in the fossil record. And so let's talk about the Cambrian cryptospores, or the so-called Cambrian cryptospores, the ones that most people, most of the paleobotanists that deal with this issue just kind of ignore, and Charlie Wellman refers to them as algal scum. So in the Cambrian, the oldest stuff we have is from the Rome Formation. There's your geology, which is uppermost series 2. This kind of junky looking material. But as of, well, just 2 years ago now, I've started just starting the systematics of describing these things systematically. So these are, this is a genus called Cisospora, and it's got these rounded, smooth-walled spore-like things. But you'll notice that here they are in fours, right? But they're not really fours. It's not just a straight classic meiotic division. This is Vidalgia, same way. This has a different kind of pattern to the surface of the wall. It's thinner, but again, you get these clusters of spore-like cells that occur. Adenosporus is another. This is kind of the basic. I'm being a lumper here, not a splitter. So this has a very wide range of morphology, and I've split out a second genus just based on this sort of pairing that you get, distinctive form. And then this Bullatus form, these are from the Grand Canyon, from the Tapeats, from the Bright Angel Shale. You get these sort of bulbils that occur in there that's pretty distinctive, and at least they make it from Arizona to Tennessee. This is work that Willie Taylor has done.
250 slices through one specimen.
There's the result, the little diagram, and if you're, it's annoying. Sets of enclosed dyads, sometimes you get 2, sometimes you get 4. So this is part of the argument. There's a long argument. It's in the literature now. These are published papers. But that meiosis is messy, and you don't really see regular divisions from a diploid sporocyte into 4 haploid spores until the middle Ordovician. Oh man, I've already got I'm not going to make it through. Laminated spore walls, plesiomorphic. So here's these tetrahedral tetrads that come in the Darwinian in the middle part of the Ordovician, and this in the literature would be the beginning of the embryophytes for a lot of people. Here's what they look like if you put all of these deposits on one slide. These guys are smaller and irregular, and there's this line that happens, and everything gets bigger and much more regular in terms of the spore characters. And then finally you have a plant up here in the Silurian. Now, one of the things so Bauer's hypothesis was that you start out with spores. The zygote produces spores, and this would then represent the spore pollen and transfer hypothesis. And then you get a thallus that is basically a spore thallus, and then eventually you have this is his quote, sterilization of sporogenous cells to form the vegetative thallus. So we have a couple of examples. This is Cambrian, where you have these regularly arranged, essentially sporocytes, spore-producing cells, that are in this case sort of a one-dimensional pattern. And then in this Dapingian sample from Fossil Mountain in Utah, we get these two-dimensional patterns, and you actually have periclinal and anticlinal divisions here to produce rosettes of these. But at the end of the day, these are not red algal vegetative cells as what's Gregor-Talic things, but they are actual spores. So that is a spore thallus. So here's some of the other examples that we can see. That we go from imperfecto triletes, which is this broken trilete spore, to actually two triletes, and these come in near the base of the Silurian. There are some patterns of cuticle-like cells that look more like they were covering plant-like tissue instead of nematothallus, which is kind of weird. And then the tubes have been not really well studied. There's lots of different structural characters associated with these various tubes, and including ones in which you have fibrillar structure in the primary wall, and in some cases these secondary thickenings are actually peeling off as a separate issue. So this is something that we really haven't addressed yet, and it's not really published on in a serious way of looking at the complicated structure. And I've just tried to point out that there's lots of different ways you might make a tube, but none of these are a good fit for what we actually see in the record. And so my argument would be that these really exhibit, structurally, they're very much like tracheids, except that they don't taper at the end, they're just tubes. But they seem to be structural components of a plant-like nature. Well, this is probably hopefully the last slide, but basically what I've tried to show you is that it is possible to literally read the fossil record in terms of plant debris and go from spores and these spore thalites and then eventually into trilete spores and then these structural components of cuticles and tubes that could be construed as parts of the physical manifestation of this genome assembly that represents eventually the true embryophytes, which is going to occur at the end of basically near the Siluro-Devonian boundary. And there's my co-author. This is a plea that this issue of the fossil record prior to the Middle Ordovician tells us nothing, and I think it probably tells us a lot. Thank you very much. And we have time for 60 seconds of questions while we do the turnover to the next. Yeah.
Just what do you mean by irregularity of meiosis? How can you have irregular meiosis and not get haploid?
Could you say that louder, Jim?
Yeah. What do you mean exactly by irregularity of meiosis? Meiosis doesn't give rise to haploid cells or extra divisions or what?
This is from, what's the guy, Haig? Work, the reduction division in streptophytic algae like Coleochaete and other streptophytes, you get endoreduplication of the genome, of the genetic material, and so you get multiple copies of the genetic material without cytokinesis, and so you end up with an irregular number of end products, which presumably are one end.
In the spore site?
In the spore site, yeah.
Okay.
And I didn't go over that, obviously, but that pattern of multiple, the pattern of cytokinesis is left over in the walls of the spore packets. And that's stuff that Willie has done, and that's also in the paper, so. Next.